Hello, I'm Rosalind Le Feuvre, the Director of Operations for the UK Biofoundry SYNBIOCHEM, which is a synthetic biology research centre focused on fine and specialty chemicals production. The Manchester Biofoundry was set up in 2014 with generous research funding from the UK Research Councils and is based at the University of Manchester's Manchester Institute of Biotechnology. The centre is set up on the first floor with significant capital funding for a suite of robotics platforms and mass spec instruments, along with the expert technical staff to support the infrastructure. Whilst our laboratories in this strange year are now up and running with social distancing, it will be so great to welcome visitors to the Institute once again, as seen in this photo of one of our open days, where we welcome 200 college students to the Institute. I'd like to start by thanking the interdisciplinary SynBiochem research team who have delivered the work I'm about to present, along with the academic leadership team. There is now a global agreement that we need to reduce our reliance on petrochemicals, and we need to find alternative, sustainable bio-based routes to chemicals manufacture. The Manchester Synthetic Biology Research Centre has created an integrated biofoundry for the production of fine specialty chemicals production based around the classical design, build, test, learn synthetic biology pipeline. In this talk, I hope to demonstrate how this now provides a rapid prototyping platform for smart biomanufacturing, offers access to diverse products for delivery at scale, and the prospect of just-in-time agile production. In creating this automated platform on the pipeline for microbial cell factories, we wish to meet a number of criteria. It would need to be agnostic to the target compound and also to the host organism, function without prior knowledge of the chemical space, and leverage high throughput automation platforms for improved performance and reduction in human error. And finally, provide software that is useful and platforms, along with the expertise for the wider synthetic biology community. This slide shows an overview of the automated design, build, test, learn workflow. From the initial chemical target, the workflow uses computational design tools for the metabolic pathway and enzyme selection of DNA parts are then designed with reusable gene blocks, and design of experiments is applied to efficiently explore the experimental design space, saving both time and experimental resources. Oligonucleotides are designed and purchased, and work lists generated for our robotics platforms for automated LCR pathway assembly, sequence verification, growth and induction, followed by quantitative screening of target product and key intermediates by mass spectrometry. The resultant data are also then used to reinform redesign of the new pathway variants for subsequent build and test in an iterative manner until desired product titers are obtained. This pipeline was first tested with 3 key classes of natural products: the terpenoids, flavonoids, and alkaloids, which have of particular interest as flavours and fragrances, but also as pharmaceutical precursors. Since this work was performed on chemical targets that were relatively familiar with us in the Institute and was performed whilst we were actually setting up the pipeline, we decided to further test the speed and the agnostic capabilities of the pipeline through a pressure test. The aim was to apply the pipeline to novel chemical spaces where we had no prior knowledge and experience, and to tackle a large number of targets simultaneously to quantify production within a relatively short time frame. The time frame would include initial target selection and the design of genetic parts and gene synthesis, and ultimately we hope that this would help us identify bottlenecks and limitations of our pipeline for future improvements. We selected targets from the top 30 bio-based platform chemicals and targets from a Shiki Factory 100, which is an EU project, many of which were identified as industrially relevant materials, monomers, or novel compounds with similar functionality. We applied this pipeline to 27 materials, monomers, precursors, and advanced intermediates across 7 different chemical classes. The targets included products used in plastics and rubber production, with the rationale that a biosynthetic route to manufacture could provide cleaner, bio-based alternatives to harsh petrochemical-based production methods. The 17 monomers from 5 of the 7 classes were successfully produced, as indicated here in green, at titers close to those reported under optimized fermentation conditions. Highlighted in blue, we also used the opportunity to engineer E. coli host chassis to boost production of primary metabolite precursors and thus increase substrate availability. By using a variety of knockouts, knock-ins, and genome and pathway integration techniques. Naturally, we did encounter a few problems along the way. Some were in the area of pathway selection. We use a tool called RetroPath. It didn't always find the right good pathways. Sometimes in the enzyme discovery step, in this case in the dicarboxylates. And we also had problems with correct assembly in a few cases and detection issues in other cases, since we had to develop mass spec methods for the quantification during the 85-day time frame. In many cases, we did identify the problems that had occurred, but were unable to go back and solve those problems within the time frame. And we have now since addressed many of these issues. However, overall, it was a big success, and with 17 of the 27 targets produced in vivo from 5 out of the 7 target classes. We designed 135 genes with 187 unique plasmid pathways assembled, verified, and tested in vivo, involving 142 unique in vitro enzyme assays. 9 mutant E. coli chassis were constructed, and 22 quantification methods were set up across our suite of mass spec instruments, analysing over 8,000 samples. We then asked ourselves if we could scale up any of these compounds to the grams per litre scale, and over the next 2 months developed a scale-up pipeline. This included directed evolution for the improvement of enzyme activity and substrate availability, pathway optimization to improve flux and cofactor redox balance, and strain, media, and finally fermentation optimization, successfully reaching grams per litre stereoselective production for both mandolate and hydroxymandolate, reaching grams and multiple grams per litre. We then decided to revisit our flavonoid projects, which had been halted due to the pipeline stress test. These are natural plant-based flavonoids which have drawn significant attention as dietary supplements due to their potential application in human health and nutrition. Currently, the industry production is either by extraction from plant sources, where there's affected by limited availability, prolonged and unpredictable crop harvesting, and multiple solvent-based purification steps which affect the yield and cost of production, or produced through de novo chemical synthesis, which is unsustainable, requiring toxic solvents and extreme chemical reaction conditions. So we focused our attention to naringenin and pinosembrin, which we had previously produced, but also to iridictol and humiridictol. And these 4 compounds sit in pivotal positions at important branch points in plant flavonoid biosynthesis pathways and provide access to a wide range of over 40 compounds. For the production of naringenin, we based our pathway on previous best producing pinosembrin pathway and selected C4CL and TAL enzymes and produced a small 15 pathway library which showed production of coumaric acids to various degrees, but importantly showed 4 of these pathways showed good production of naringenin in reasonable amounts, And this production was increased by the addition of cerulenin. Those pathways that didn't produce neuroenginein had obviously had some problem along the way, because we did get a production of chimeric acid in many of these. We then tested the 4 best pathways against our common E. coli strains and found that DH5 alpha showed the best production at about 250 milligrams per litre, and reaching up to 300 milligrams per litre. This was then tested with different growth media and carbon sources, reaching a titer of 350 milligrams per litre directly from glycerol. At this point, we still needed to add the expensive additives tyrosine and cerulenin to achieve our best titers. So we then addressed the questions: can we remove the need for these additions by engineering our strains? Looking at the addition of cerulenin, which significantly increased the production in our base strains, we used a multiplex CRISPR-I system and demonstrated that through genome editing or gene regulation, we managed to alleviate the need for adding cerulenin and achieved good titers in the absence of the fatty acids inhibitor, reaching to 350 milligrams per litre. And then moving on to tyrosine, we'd previously demonstrated a gene knockout, a double gene knockout, which was able to boost the metabolic flux towards tyrosine overproduction and restrict phenylalanine production. And we improved this with a tyrosine shunt pathway to develop a strain capable of producing 500 milligrams per litre directly from glycerol. We were able to then troubleshoot our ferulic acid pathway that we'd used in the pressure test, and realised that one of our genes was actually incorrect, and went back and constructed a pathway producing 200 milligrams per litre. So we're then able to move from glycerol straight through to ferulic acid. By combining this with the best conversion pathway that we had demonstrated in the production of neuroenginein, we then were able to show the first production of both homoeridictol here and eridictol, admittedly at low levels. So I hope I've managed to show a couple of experiments of how we're able to deliver quick access to diverse chemicals and prototype microbial production strains for scale-up and optimization. To deliver benefit of the synthetic biology pipeline, SYNBIOCHEM Foundry is now working with several other centres, and I will just quickly touch on a couple of these. The Henry Royce Institute for Materials Science is working with us to extend our work on materials monomers for materials from biology, and the Future Biomanufacturing Research Centre is working with us on scale-up of our production strains. So whilst most of our automation has previously been performed on individual Hamilton star instruments with plate readers and incubators, we have just installed a fully integrated robotics platform on the first floor, which was funded through the Voice Institute. This platform integrates acoustic and pipette-based liquid handling robots with the necessary centrifugation, plate readers, washers, plate sealers and de-sealers, PCR instruments, and incubators. And this platform is aimed at the rapid discovery and engineering of advanced materials from biology. And we're now setting up fully automated directed evolution and synthetic biology protocols on the platform. We were also fortunate enough to also gain funding for next generation sequencing, which then becomes a bottleneck when you increase the numbers, and automated colony picking, and also luckily through further investment from EPSRC on mass spectrometry, which will allow us to do high resolution but also high throughput mass spectrometry to help alleviate the bottleneck when you start looking at thousands of samples the analytics is likely to become the bottleneck. Finally, we are working closely with the Future Biomanufacturing Research Hub, which is also based at the University of Manchester and links academic spoke partners at University College London and Imperial College London, University of Nottingham, with scale-up facilities, including iBioIC and CPI, and industry. The hub is addressing two main challenges that need to be addressed for biomanufacturing to become a reality: of speed of biocatalyst engineering and the cost of biomanufacturing at scale, which need to become for this to become economically viable. The Biofoundries is also particularly working to develop high throughput analytical platforms for screening large numbers of constructs, and industrial host train engineering, where this will really help in biomanufacturing overcome the economics of manufacturing at scale. This will hopefully include reducing the reliance on freshwater consumption, the use of renewable feedstocks, ideally using non-sterile conditions, or at least to reduce the need for antibiotics and reduce hazardous waste byproducts. So I'd like to just finish by thank you very much for listening, and thank you very much indeed. Bye-bye.